Okay, welcome to NGS Module 1, NGS now NGS Online Workshop. So, yes, Priyanka, so from your side, how much exposure do you have to NGS data analysis?
I'm not using any of the NGS tools. I'm just using drug discovery,
Okay,
using bioinformatics tools. I'm learning it now.
okay. So, your major area of research is in?
My area of research is in bioinformatics and lung cancer, like drug repurposing. So I have joined the PhD right now.
Okay, so as you know, data quality is common to all the NGS sequencing. And this is the first module. It's common to almost any kind of sequencing. Dr. Party has joined, so did he join? Okay, there's one more participant. Okay, let us wait for him. Meanwhile, I will close this and again screen we lost, so I will share screen again. So what do you see here? The instructors of this manual. Yes, the trainers are okay. So to this is mm, yes. What does the title say? Yes, Priyanka.
Introduction to Next Generation Sequencing, hands-on workshop.
Yeah. So this is a good starting material, but still, before going to this, I will go to another PDF where we can have a theoretical overview of NGS terminologies. So these are the instructors of this manual, mostly postdocs, as you can see, mostly postdocs and some early career scientists. So these are the types of legends here. Okay, if it is marked with violet, I mean dark purple, it is important. If it is green, notes, it's for reference. Light green with footsteps, it's follow the steps. It's yellow Q, it's questions to answer. It's red exclamatory, it's warning, stop and read. And then green octagon, star octagon, it is bonus exercise for fast learners. And blue star octagon, advanced learning exercise for fast learners. So, module on data quality. What are the key learning outcomes of this module? Hello.
Yes, sir.
Yeah. What are the key learning outcomes of this data quality module?
To assess the overall quality of NGS sequencing, visualize the quality and other associated metrics of which to decide on filters and cutoff for cleaning up data ready for upstream analysis, and to clean up and preprocess the sequences data for further analysis.
So, these are the tools we will be mainly using: FastQC, FastXToolkit, and Picard, and FastQ encoding. Also, you must know. If you open this link, so data quality requires an understanding of the FastQ format of raw reads. So we will first deal with some basic terminologies. Yes, please. These three main workflows on three columns. What are the three possible analysis we can do? This is
Variant validation
this is.
consensus assembled genome and
And
de novo assembled genome.
so for variant validation, what are the steps?
uh, G- Genome database to FOSS sequencing, reference genome sequence, then mapping reads to reference genome, then detailed information on coverage, read, depth, and placement, SNP summary and insertion deletion, annotate variants, and then finally variant validation.
So this is not SNP. So this is as we see here, chat box. Yes, how do you pronounce it?
SNIP,
Snip. Okay, snip.
SNP.
Okay, yes. Pronounce it as snip. And coming back. Yes, okay. Party, please join.
Yeah.
Yeah.
The same link, right?
Yeah.
Okay.
So, for consensus assembled genome, what are the steps?
FOSS reads uh, sequence reads, reference genome sequences, mapping reads to reference genome, detailed information coverage on coverage rate, depth, and placement. Yes.
Okay, for de novo assembled genome, what are the steps?
Can you just go
Yeah, okay.
de novo config assembly, build scaffold, align scaffolds to chromosomes, annotate genome, and then de novo assemble the genome.
So, for both for variant validation as well as consensus assembled genome, we require genome database, right?
Yes.
Yes, and also we require from the genome database what we require, the reference genome sequence. But for de novo quantic assembly, directly we do it from the FASTQ reads. Okay.
Okay.
Yes. So first of all, we need, at least, do you know about FASTI format? Yes.
Yes,
You,
sir.
yes.
Yes.
So let's revise what is FASTI format here. Yes. What is FASTI format?
A widely adopted sequence format used for protein transcript or genomic RNA or DNA sequences, often marked by one of the following file extensions: .fa, .sna, .faa, or .fasta.
Yes. Please continue.
In the simple flat file format, entries begin as a single descriptive line denoted by greater than symbol, followed by a hard return, and then the sequence. Most sequence records housed at NCBI can be displayed or downloaded as FASTA formatted files. Records in nucleotide or protein databases can be changed to FASTA and pasted or saved to a local directory using the send to menu. Larger files such as chromosomes can be downloaded from FTP directory.
So it is just like SNP is pronounced SNP, right? Yes. Sorry.
Yes, sir.
Yes. So let me close this. So by mistake it opened. So just like SNP is pronounced SNP, we pronounce fast like this. Okay. Why is it hanging? Yes. How do we pronounce it?
Fast A.
Fast I. Okay.
Oh, I.
Fast I. Okay. We pronounce SNP as SNP, FASTI as FASTI. Okay, it is clear. And FA or FASTA are equivalent, FASTI, FA are equivalent. FNA is usually used for DNA FASTI sequences. Okay. Okay.
Yes, sir.
Yes, FNA is usually used for DNA sequences which are in FASTI format. So FAA is used for if FNA is going to be used for DNA FASTI sequences, FAA is going to be used for protein sequences. Okay.
Okay.
So that's what it's given here, right? Proteins in records in nucleotide or protein databases, right?
Yes.
Yes. So now coming to FASTQ. Yes, what is FASTQ?
The most common raw, unaligned data format for next-generation sequencing marked by the file extension .fastq. This data format output from sequencing platforms includes sequence data and the quality score for each position. FASTQ files can be dumped from SRA using the SRA toolkit. If the SRA record is aligned, a BAM or submitted, FASTQ files comprised of reads that correspond to a given genomic region can be dumped using the SAM dump utility.
So just like FASTI can be represented in .fa, right?
Yes.
FASTQ can be also represented in,
Dot fast.
that's what, dot FASTI dot fa. Similarly, dot fq. Okay.
Okay.
Good evening, Dr. Padi.
Yes, good evening. I'm so sorry.
No problem. So this week we are going to cover data quality. Okay.
Okay.
Because that's anyway important for metagenomics also. So
Yes, okay.
data quality control.
Yes.
So we were discussing about basic things like SNPs and FASTI format and FASTQ format.
Okay.
So we have with us a participant, Priyanka Ramesh is pursuing a PhD in VIT University Vellore. So she's working on lung cancer. She just started her PhD. So Dr. Priyanka, this is Dr. Srichandan Padi, is MK-BAN Young Research Fellow at Institute of Biosciences and Sustainable Development, Sikkim. So he has also been a visiting scientist in Italy, Naples, for some time. He works on environmental metagenomics. Okay, so let us come to simple terminologies. What is BAM file? Yes, Dr. Padi, what is BAM?
BAM.
Yeah, I highlighted it, right?
Okay. Do you want me to read?
Yeah.
Okay. Binary files containing reads aligned to a reference genome marked by the extension .bam. Currently considered to be the universal file format for next generation sequencing. These files are made up of reads mapped to a genome. SAM dump, part of the SRA toolkit, can extract any portion of aligned SRA data from NCBI, potentially making downloaded files much smaller. SAM files can be converted to BAM using SAM tools. BAM files are used for many things, from variant calling to RNA seq counts and isoform detection to epigenomic recalling.
Yeah, so BAM full form is
Binary
binary aligned map format. Okay. Yes, so BAM file is nothing but as we can see here, BAM file is nothing but FASTQ plus yes. What is BAM file? I wrote in chat. Yes, Priyanka.
Yes, so BAM equal to FOSQ plus alignment information to reference FOSA cell.
So it is possible to convert
BAM file to SAM file.
no. SAM file should be converted to BAM file, not other way around. It's possible to convert BAM to FASTQ. Okay. Because we are only discarding some information. What information we are discarding? We are discarding the alignment information, right? Then BAM file is nothing but FASTQ file. In order to convert in order to obtain the BAM file from FASTQ file, we need the reference genome. Okay.
Yes.
So, what is coverage?
It is not visible in your screen.
My screen is
Yes.
big enough.
Yes. Coverage. You want me to read or
Yeah.
she can read?
Okay, Priyanka, please read.
Genome coverage, depth of coverage, the average number of times each base position is sequenced. Coverage can be simply calculated by number of reads into read length by genome size. GOSRA and WGS whole genome sequence databases at NCBI contain this information. When calling variants or calculating read counts in a particular region, coverage data is very valuable in calculating the reliability of it.
So, GEO is gene expression omnibus, SRA sequence seed archive, WGS has mentioned their whole genome sequence. So, coverage is number of reads into read length divided by genome size. Yes. So, as per definition, average number of times each base position is sequenced. So, suppose this is a read position, okay. Let us say this is or let us say yes. So suppose this top rows, right? Top rows, let us say, no, don't consider the bottom squares, only the top 3 squares you consider.
Okay.
So from right to left, let us say this 3 length is a read length, okay. That is this 3 length sequence is a read, and let us say white is 0, black is 1, okay. White is 0, black is 1. Is it understood, Priyanka? White is 0, black is 1, okay.
Yes, sir.
So we have white, 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 black, white, black, white, white, black, 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 white, white, black, white, black, 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 white, and black, 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 right? Yes. Can you tell that in terms of zeros and ones?
111, 110, 101,
Yeah.
100,
Yeah.
011,
Yeah.
010,
Yeah.
001, 000.
So I am writing that in a Word document. Let me before writing that in a Word document, let me just open this publication. So my own publication in Asia Pacific Bioinformatics Network Singapore.
Okay, for you now. How you manage? Okay, fine. Then lunch what you're told it?
So
Yes. Then eating some pakoda or something like that.
let me download it. Yes. So these are what is the 8 strings, right? Yes. 111 is, instead of being on the left, it's on the right, right? Yes.
Yes, sir.
So 000, 001, 010, 011, 100, 101, 110, and
Triple O.
so let me establish that this is the reference genome. Okay, these are the 8 reads. Okay, these are the 8 reads.
Okay.
What is the specialty of these 8 reads? These 8 reads are all the possible 3 length reads we can have, right, in binary. Yes. In 3 length binary, these are all the 8 combinations, right?
Okay.
Yes. There is no other combination. These are all the possible 8 combinations. Yes.
Okay.
So now let me say this is the reference genome. Okay. Yes, Priyanka, what is the reference genome?
0001011.
And what is the position? This is starting from 1, right? Index position is
Yes.
starting from 1, right?
Yes.
So Dr. Padi, what can you say about yes? What is these 2 types of numbering, Priyanka? 0 based?
0 based numbering and 1 based
One
numbering.
base number.
Yes. So what is 0 based numbering, Priyanka?
0 based numbering is the computational idea of indexing an ordered data structure, example a string or array, by starting with 0 instead of 1. For example, if S is the string ACGT, then S of 0 is given by the symbol A and S of 1 is T.
S of 2 is
G.
S of 3 is
T.
T.
so in 0 based numbering the index goes from 0 to length minus 1, right?
Yes.
Okay.
Yes, simple. So 0 based numbering is
0 based numbering is used by default in a wide range of programming languages including Python.
okay. So Dr. Padi, already you read it, but let us repeat it. This is very important in bioinformatics numbering. So 1 based numbering, Priyanka.
1 based numbering is the computational idea of indexing an ordered data structure, example a string or array, by starting with 1 instead of 0. For example, if S is the string ACGT, then S of 1 is given by the symbol A and S of 2 is T.
S of
By
3 is
default, G,
S of 4 is
T.
so in 1 based numbering index goes from 1 to L, right? Length, right?
Yes.
So by default anyway that's
Problems in Rosalind's bioinformatics stronghold will use 1 base numbering instead of 0 base numbering, which is used by Python.
yeah. So in Python we are using 0 based numbering, and here
1 base.
1 based number. But anyway, whenever you have a sequence database in bioinformatics, you must be very careful about the numbering involved. Okay.
Yeah.
Because even one position change can alter things, right? So
Yes.
so fundamentally, why these 2 types of numbering? one base numbering is natural numbers, right?
Yeah.
Yes, what are natural numbers, Priyanka? 1, 2, 3, 4, 5, 6, 7, 8, 9, right?
Yes, sir.
And zero base numbering is whole numbers, right?
Whole number.
So that is why we have fundamentally one base numbering in natural numbers encoding and whole zero base numbering whole number encoding. Very simple. There is no point in having two base numbering and so on, because again why 0 and 1? Because the minimum prime number is 2, right? And anything to the mod 2, any number to the mod 2 is either 0 or 1, right? 0 or 1, right? So yes, mod 2 is the minimum mod we can have for having inequivalent remainders, right? Mod 1 will give you always equivalent remainders of 0, right? Yes, anything you divide by 1, remainder will always be 0. 2 is a minimum number which will give you two different remainders.
Yes.
And two different things are enough to compare, right? So, okay, let us not go into much intricacies of that. Let us come back. So, this screen is not visible now.
No, sir. It's
Okay,
not visible.
okay, I'll share the screen. So that additional pop-up was for screen. So I thought it was some other. Okay, so coming back to coverage, why did we share this work? So fundamentally, here I am saying I'm randomizing 8 strings of in binary, and where is that PDF? So I downloaded it here. Or I downloaded it here. Yeah. So 0 0 0 0 0 0 1 0 1 1 1 I claim to be the reference genome for this 8 reads. Okay, we will verify that. So is 0 0 0 contained here?
Yes. First.
In which position? 1 2 3 position, right?
1, 2, 3.
Yes. Yes.
Is 0 0 1 contained?
2, 3, 4.
2 3 4, right?
4%.
0 1 0 is there?
3,
3,
4,
4,
5.
3 4 5, right? So these are all forward mappings, right? Forward mapping. Yes.
Yes.
So is a 0 1 1 contained?
0 1 1 5 6 7.
Yeah. So now we come to the first reverse mapping. 4 3 2 is reverse, right?
Yes.
Yes.
4,
1 0 0 is in reverse orientation, right?
Yes.
1 0 1 is in both orientation. 1 0 1 is symmetric, right? 1 0 1 1 0 1. 4 5 6 4 5 6 and also 6 5 4, right?
Yes.
And 1 1 0 1 1 0 all reverse, right?
Yes, sir. Reverse.
Reverse, right? 7 6 5. 1 1 1 is
Both forward
both,
Six,
right?
seven,
Both
eight, eight,
forward,
seven,
both
six.
forward. So 0 0 0 is also both, right?
and yes,
Yes. 1 2 3 3 2.
So how many both are there?
3.
3 or 4?
1 0 1 1 0 1 triple 1 and 0 0 0.
Yes, 0 0 0 1 1 1.
Yes. 1 0 1.
1 0 1 and also 0 1 0. 4
0, 1,
4,
0.
4 both, right? 4 both.
Yes.
So if I'm going to count anything under both, then I don't count them again in forward and reverse. Okay, is that clear?
Okay. Among them, only one will be counted.
No. If I'm going to count, if whatever 4 both I have counted,
Yeah.
I am not going to count them again in forward and reverse. Okay. So how many forward and how many reverse? 2 plus 2, right?
2 plus 2. Yes.
Yes. This is 2 3 4. 5 6 7 and that is 2 forward and 7 6 5 and 4 3 2. So we see that both are basically symmetric and forward and reverse are asymmetric, right? Yes, whatever is both, they are symmetric, palindromes, right?
Yeah,
Yes,
palindromes.
sir.
Yes.
And whatever are forward or reverse are non-palindrome. So 4 plus 2 plus 2 is 8.
Eight.
Yeah.
Yes, so is it convincing that this is a reference genome?
Yes.
Yes, sir.
Yes. So now in this reference genome, let us come to this definition, very important. So perfect coverage, yes. What is the definition? Perfect coverage?
In fragment assembly, having a read or gamers that begins at every nucleotide.
Is it satisfied here? At every nucleotide we are beginning a read, right?
Yes, sir.
Whether
Yes.
it be forward or reverse, that doesn't matter, but we are beginning a read, right?
Yeah.
Yes, sir.
Yes. So this is perfect coverage, and that is a title. What is a title?
Some interesting observations upon perfect coverage had in the assembly of exhaustive randomized genetic studies.
Okay, this was presented by us at INSIB, International Symposium on Bioinformatics, which is conducted by Asia Pacific Bioinformatics Network in every December of every year. Okay, so in 2020 it was conducted at Vignan University, Guntur. Okay, fine. So it was online mode, and so now what is the read length?
8.
Read length.
3.
Read length is 3, right?
Yes,
Yes.
Yes. And what is the genome size?
8.
Genome
8.
size 8. 8 reads.
And what is the number of reads?
8 reads.
8 reads of length 3.
Yeah.
8 reads of length 3 divided
Yeah.
by 8 genome size. What is 8 into 3 by 8?
3.
So what is coverage?
3.
3.
Coverage is 3, which is in bioinformatics terminology, we read coverage as 3x. Okay. So Priyanka, you are working on lung cancer, right?
Yes, sir.
So for cancer, the suggested coverage is 60x. Okay.
Okay.
So not this. Suggested coverage for cancer is 60x coverage you should have. So cancer samples, I mean to say.
Irrespective of type of cancer, sir.
That is, I mean, I'll show you one IBM document. Okay, IBM
Okay.
one IBM document. So whether I should see whether that is still available. Yeah. So let it open. So it's all in one day medicine cancer patients infographic.pdf, as you can see. So in cancer treatment, right? In cancer treatment, yes.
Yes,
Let's check for coverage quickly. Coverage is 30x. Okay. For optimum results,
Yes, it is there. 60x coverage.
yeah. Is it? Where is this?
Okay.
Yeah. So what is the fourth point, Priyanka?
for optimum results, Illumina recommends a minimum coverage of 30x for normal tissue and 60x coverage for tumor samples. Whole genome sequencing at 30x coverage is approximately 200 GB of data, and 60x coverage for further tumor samples, it would be approximately 400 GB. If the patient is sequenced at least 2 times per year,
Fine, all that is same, all that is fine. So you will see that for a control data set it is 30x, for case data set 60x, right? So
Okay.
yes, so that is fine. So this is from Intel. Fine. So if you want, I will share that link later in email. So fine. So coming back to this, what is the interesting observation? I said interesting observation, right? So it is
yes,
first of all, coverage is 3, 8 gets cancelled for randomized strings. But what is the interesting observation? This, if you see in Rosalind, so if I come to glossary, Hamming. Yes, what is Hamming distance? Yes, Dr. Paddy.
Yes.
Yeah.
The Hamming distance between two strings, Hamming distance between two strings having the same length is the minimum number of symbol substitutions required to transform one string into the other. If the strings are given by S1 and S2, then we write the Hamming distance between them as this is derivative or delta
DH.
dh s1 s2. We can compute the Hamming distance by visual inspection. The Hamming distance between two strings is simply the number of positions in the strings at which corresponding symbols differ. For example, the Hamming distance between the two strings is the figure below must be equal to 7.
So Priyanka, what are the red symbols and what are the black symbols?
G,
No. Why red and why black? What is the difference between red and black encoding?
Substitutions.
No, red is just mismatch and black is match, right?
Yes, yes,
is
yes.
So number of mismatches is Hamming distance, right?
Yes, yes.
Yes. So potentially, if you have two strings of same length, minimum Hamming distance will be, can be minimum Hamming distance will be.
Sorry, I couldn't get.
If you have two genetic strings of same length, minimum Hamming distance will be.
Minimum same minimum 1.
Is it 1 or?
Minimum, na?
Yeah. If the two strings are the same, Hamming distance will be
0.
yes.
0.
Is it okay, Priyanka?
is
So let us say I have, for example, in the meet, I have these two strings. Yes. What is the Hamming distance between these two?
0.
0, right?
0.
So now the Hamming distance between second and third is
Almost 1, 2, 3, 4, 5, 6. Entire length.
entire length, right? So minimum will be 0 and maximum will be entire length, right?
Yeah.
Yes.
So what is the interesting observation that we made here? Where is the PDF? Okay, PDF. The interesting observation is among these 8 strings, maximum Hamming distances between 0, 0 and 1, 1, 1, right?
Yes.
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 So what is that maximum Hamming distance? Between okay, we'll go systematically. Hamming distance between 0, 0, 0 and 0, 0, 1 is
1.
First and third is
First and third. Also 1.
1,
first and fourth is
2,
Uh, yes, 2.
first and fifth is
1,
1.
yeah. Sixth is
2,
2.
seventh is
2.
2,
eighth is
3.
3,
and what is coverage?
Coverage is 3x. 3x.
So the interesting observation that we made is if we assemble perfect coverage of randomized strings, potentially coverage will be equal to maximum Hamming distance. Okay.
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 This is a fundamental result. It has a very fundamental result. So this has been validated by me only for this type of strings, binary 3-bit strings. But what I am today only I was sitting and doing using this random sec. Okay, using this random sec, I was generating random sequences and assembling them. Okay, fine. So anyway, coming back, so we will go back to these 4 definitions we saw. Is coverage clear now?
Yes, clear.
According to definition, the average number of times each base position is sequenced. Okay. Yes. So in this I am writing a Word document. So I am opening it in big font, 40. Is this clear?
Yes.
Yes.
Yes, sir.
And then one zero, right?
Yes.
How many reads cover? 1, 2, 3, 4, 5, 6.
6.
How many reads more left?
2.
What are the 2 missing reads? 101, right?
101 is there. 101 is there.
101 is there.
011. 011 is also there.
Yeah. 011. 0. Oh, that is 110. That is reverse. Whatever is up is reverse. Okay. Whatever is up is reverse and whatever is down is forward. Okay. Okay. So quickly tell me what are the others. Reverse. What are the others? 0, 1, 0.
110 is there.
Yeah.
101.
101. Yes.
Then 010.
010.
Yeah. 010 forward I did down, right? So
Yes.
101. 101, yes.
010.
So 010. 010 I wrote here, right? Here.
Also 101 you have written.
Okay, 101 I wrote there. I'll again let us check with the reads.
Yes.
000, 001, 010, 011, 100, 101, 110, 100 and 110. 100 and 110. 110, 100, 100. Yes, 100. Yes, 8 done. Are we
Yes,
repeating?
No, also, okay, I understand. Yeah.
0 0 1 0 1 0 1 0 1 0 1 1 0 1 is repeating.
Repeating, right?
Repeating. So what is the other one? Yes, Priyanka, what is the other one? 1 0 0 0 0 0.
One.
We'll check one by one. 0 0 0 0 0 0. Okay. 0 0 1 0 0 1 0 1 0
Yes,
0 1
there.
0 1 0 1 0 1 0 0 1 1
Yes,
0
there.
0 1 1. This is 1 1 0. This is not 0 1 1. Okay, this is 1 1 0. So 0 1 1 I should write here, right? Yes, now is it fine? This is whatever is up is reverse, okay? Please.
Yeah.
Yes.
111, 110, 101,
Yeah.
100.
Yes, sir.
Reverse. This is reverse.
Then 0110.
0 1 0 is forward, right? Whatever is both I can write up or down, that doesn't matter, okay?
Okay.
So whatever is both I will write up or down, that is my choice, but whatever is up is definitely backward and whatever is down is definitely forward, okay?
Yes.
So now what is the average number of what is coverage definition here? Average number of
Average number of nucleotide
is average number of times.
times each base position each
Is within a
sequence.
genome.
Average number of times each base is sequence. So 0 is sequence average how many times? What is the read correspond how many times? One time only, right?
0, yes, 0, one time only.
Yes, again 0 is sequence 2 times, right?
Yes, sir.
Yes. So let me write here, please. 1 plus 2 plus. Yes, tell me quickly. Quickly tell me. 1 plus 2 plus. Is it
1.
is it 2? Here is it 2? Is this 2?
So 3, sir.
It's 3, right? 1 plus 3 plus
1, 2, 2, 3.
1 plus. You should cover both backward and
4,
forward, right? 1 plus 3 plus.
4,
Yes. 1 2 3 4. Yes. 4 plus.
4,
Yeah. 1 2 1 2 3 4. 4 plus.
again 4,
Again 1 2. Yes. Are you counting this position?
yes, sir.
Yeah. 1 2 3 4, right? 4 plus. Yes. Now
Again 4,
4 plus
3,
3 plus.
1.
What is the total? 3 plus 1 is 3 plus 1 is 4, 3 plus 1 is 4, and we have 4 fours.
24.
It's 16. 16 plus 4 plus 4.
4 plus 4.
24, right?
24.
So 24, average number of times each base pair is for 24. What is the genome length?
8,
So what is the average number of times it is sequenced?
3.
24.
That is a coverage that we got, right?
Yes,
Yes.
sir. 3.
So now if you compute, what are we counting when we are counting the average number here? We are actually counting number of reads into read length, right?
Yes,
Yes.
Number of reads into read length. How many times we are counting? That is number of reads. This is 1 2 3 4 5 6 7
8
8
times.
8 reads we are counting.
Yes.
Yes, this 4, okay, this 4, if you write as 3 plus 1, okay,
Okay.
yes, and this 4, if you write as 3 plus 1, this 4, if you write as 3 plus 1, this 4, if you write as 3 plus 1, yes, so that 1 plus this one, this one, this one, this one, this one, this one, and this one 6 ones, right?
Yes.
6 ones will become 2 threes, right?
Yes.
And the remaining are 6 threes, right?
Yes.
So it's as good as doing number of reads into read length divided by genome size, right?
Yes.
Did you understand? Yes.
Yes.
Yes,
Yes.
sir.
Yes.
Yes. So see, this is a way to learn the basics of NGS. We cannot directly do some analysis and then think that we are doing something big, but nobody would have taught you coverage concept like this. So fine. So we will uh, we will meet Tuesday 7 p.m. Yes.
Okay. Okay.
Okay.
Yes. Yes.